this was to test for carriers of sickle cell disease, because there was a restriction site within the beta globin gene that is the cause, basically, mutated beta globin gene that causes sickle cell disease, how they were able to identify carriers. And I was just getting ready to start talking about the second medical test. That was another early one that was used with restriction enzymes. And that's the test for Huntington's chorea. And so that's what we'll pick up today. Now, Huntington's chorea is a neuromuscular disease, and it generally develops after an individual has turned 40 or in their 50s. And once it starts, it progresses on to death. Basically what happens is the first things to be affected are anything associated with skeletal muscle, and then some of the smooth muscles are affected there. And so individuals lose control over their muscular system, and then they ultimately lose control of their ability to talk, their ability to breathe, and so on, and it is failed. The mutation in the Huntington's disease that results in the development of Huntington's chorea is a dominant mutation, not a recessive one. And therefore, anyone that has just one mutated gene will develop Huntington's in later life. And because there at that time was no way to identify carriers. Well, there aren't carriers. I mean, either have it or you don't have it, basically. There was no way to identify individuals that would develop Huntington's. So, in the mid-1980s, mid-late 1980s, a faculty member at UMass in the psychology department, is PhD in psychology, her name is Nancy Wexler. Her mother died of Huntington's. And she and her sister, therefore, had a 50% chance, dominant gene, a 50% chance of developing Huntington's in their later life. And she was concerned about that. And so she went to an individual who was at Mass General Hospital doing research. And she said, is there any kind of a test that you think possibly could develop to identify individuals that will develop Huntington's in later life? So if they choose to have the test and it comes back that they will develop Huntington's, they will have the ability to make decisions about childbearing because, of course, if a person develops it, then they can pass that on at a 50% possibility for any child that they are the parents of. And the group at Massachusetts General Hospital said, well, we don't know, but we'll give it a shot. So what the researchers did is they looked at the world occurrence of Huntington's. And they discovered that in isolated human populations in the mountains of Venezuela, there were populations that had there's actually a higher incidence of Huntington's than the general population elsewhere in the world, including the United States. And besides having a higher incidence of Huntington's developing in individuals in those populations, they also found that Huntington's could develop earlier in life, and they saw that in the population as well. Some individuals as young as 17 and 18 years old were symptomatic for Huntington's. Okay? So they chose to go to those isolated regions of Venezuela and explain to the people what they were doing and ask for permission for two things. One, to draw blood samples from each of the individuals that was doing, and second, to take a complete family history so that they would know what relatives died of Huntington's and so on. And they got permission, and they did that. They collected the blood samples, and they brought them back to UMass, put them in the -80 freezer, where everything goes, and started on that. But they also had a plan in place to go back periodically, so that with the family histories and the blood and so on, they could see individuals who were not expressing Huntington's when they first collected the blood, but who developed Huntington's within the next 5 or 10 years. And so they would go back periodically and add to the family history information. Okay, so they brought all that blood back to Massachusetts General Hospital, and they started on the project. Now, they were not looking for the Huntington's gene, and at that time nobody knew where it was, what chromosome it occurred, or what at the DNA level appeared to be causing the Huntington's. Okay? What they were looking for was a marker, not the gene. It's just a way of saying if you have this, you will develop it, okay? But it isn't the gene. And so what they did was they started isolating the DNA from the various samples. And of course, since it was blood samples and they were looking for DNA, they had to look at nucleated blood cells. And so they were isolating the DNA from the white blood cells. And then they just simply opened their freezer and started on restriction enzymes. They went from one restriction enzyme to the next restriction enzyme to the next restriction enzyme, and they did a whole series of digestion, and they would run the digested products out on a great big vertical gel like the old original sequencing gels, looking for a band that was unique to individuals who had Huntington's or who developed Huntington's. So looking for the carrier. And amazingly enough, considering it was just, a shot in the dark, amazingly enough, they found a marker for Huntington's at about enzyme number 23, which is really pretty quick. And so, I don't have the original data, but I can give you an idea the kind of thing that happened. Okay, these are just individuals. Individual 1, 2, 3, 4, 5, and 6. Let's say that individual number 2 and individual number 6 already were displaying the characteristics of Huntington's. None of the other individuals up there were, okay? And so they would load the gels and run out the restriction digest. And maybe they get something or other like this. If you look up here, there is a band that is unique compared to the other individuals or most of the other individuals. That is unique to the 2 individuals up here that already were displaying the characteristics of Huntington's chorea. There is also another individual, this individual, that has that band as well. But at the time, that individual was not displaying the characteristics of Huntington's. And so, this is why they were going back and doing follow-up with this. And at one point, then they went back, and individual number 4 had started to display the symptoms as well. So they had their marker. Not only, as I said, was this population having a higher incidence of Huntington's, but these populations in Venezuela were really the only ones where Huntington was displaying very early in a number of individuals. Like I said, 17, 18, 19 years old, which is really, really unusual. So, they had their marker, and that's when genetic test for Huntington's chorea became available to the general public. Interestingly enough, Nancy Wexler herself chose not to have the test. And I'm not sure why. Her sister did have the test, but it came back inconclusive, and she chose not to be retested. When you think about it, I mean, I have thought about it, I'm not sure at 21 I would want to know I was going to develop a debilitating disease in my system. That would be really kind of a heavy weight to be carrying around those things. But anyway, that was where it was. Until 1994. In 1994, the gene that produces the protein, the protein is what goes bad, and then results in the Huntington's characteristics. The gene for Huntington's chorea was identified. Its location was found within the human genome, and that was really partly because of the Human Genome Project. So all of you need to recognize that all of us have this gene, because it is a gene for a brain protein, okay? And it is a necessary brain protein. But clearly, not everyone develops Huntington's chorea. So what was going on with the individuals with this normal, in quotes, gene that all of us have, and yet they developed this disease? When they sequenced Huntington's chorea, the Huntington's chorea gene, and by the way, it was found on human chromosome number 4 near the telomere end of that chromosome. But when they sequenced it, what they found was near the 5' end of the coding sequence, and I have the coding sequence, and I even scanned it and was going to project it for you, but you can't read it, so what's the point? Anyway, you can't read it because all the letters are together. Anyway, near the 5' end of the coding sequence for that gene is a 3-nucleotide repeat that repeats over and over and over again, and this is in everybody, okay? The sequence of the repeat is CAG. CAG codes for the amino acid glutamine. And where this repeat sequence occurs, the CAG repeats over and over and over and over and over again. Typical number of CAG repeats in the Huntington's chorea gene is around 17. Okay? What they found was a correlation between the number of CAG repeats and the development of Huntington's chorea later in life. Individuals that develop Huntington's later in life have 25 or above CAG repeats in that section of the gene. And those young people that develop Huntington's chorea in their teens have over 36 CAG repeats in that gene. So it has something to do with the number of CAG repeats that is then expressed in the number of glutamine amino acids that are incorporated into that brain protein. It probably affects the way the protein folds at some point. Why the symptoms are delayed, I have no idea. And I'm not sure they even know that yet. But that is how restriction enzymes were first used in some medical tests. Now what they do is they still use the restriction enzyme approach if you've decided to get the test. But they use that as an initial screening, and then if it comes back inconclusive, they will go in and determine the number of CAG repeats you have in your Huntington's gene. That is what is going on with some of that testing. Any questions about that? Okay. So, not next week, but the week after next, we're going to do some DNA isolation in lab, and I want to talk to you in general terms about approaches for isolating DNA. The whole theory of DNA isolation is obviously pretty straightforward. we use a lot of the same chemicals in RNA isolations as well. But I want to warn you early on that you really need to know your organism that you're going to isolate DNA from, because not all organisms allow the same approach to DNA isolation. For example, I worked or have worked over the course of my career with a marine brown alga. And it's obviously photosynthetic. It's a really interesting organism. And you can isolate DNA from it until you're blue in the face, but you can't do anything with the DNA once you get it out if you don't recognize what is going on with the organism. The organism's name is Cytocycline longitaria, and it's, as I said, a brown alga. It grows about yay tall. It's like a string of sausages out in the title, subtitle. So, and as I said, you can isolate DNA, and you can get gobs of DNA out of that thing. But if you don't know the fact that it's high in phenolics, and phenolics bind to DNA and make DNA unrestrictable if they're bound to DNA, doesn't do you any good to isolate DNA, okay? You've got to get rid of the phenolics in the process. And if you don't know phenolics are there, then you're going to have a problem with the isolation protocol. The other thing that I would say about DNA isolations relative to lab work is that there are all kinds of DNA isolation kits that you can buy for biotech companies, and I don't isolate DNA that way. And that's because any kit you use, whether you're isolating DNA or RNA, any kit you use is a black box. You put in buffer A, and then you do something. And then you put in buffer B, and you do something else. You have no idea what buffer A is doing, nor do you have any idea what buffer B is doing, right? And so that works fine. Who cares what it's doing, as long as you get what you want at the end, and you can just go where you want to go. But let me tell you, it doesn't work that way in a functional lab. And if you work with something that is a black box, and it doesn't work, how do you troubleshoot the problem? You can't, okay? So I'm not big into the, let's get a kit. Anyway, some these kits have some of the same ingredients, or the chemicals that I'm going to talk about. They oftentimes, however, say it's proprietary and they don't tell you what it is. And so again, you have a problem if the isolation did not work. I'm not saying here, so don't misquote me, I'm not saying here that you should never use kits. I have used kits as well. The kit is going to work really well. Get what you want. Get it purified and in a form that's functional. Go for it. But no, just always know when you make a decision to go with the kit, if you have problems with it, and there are problems with every kit, you will not have an easy time troubleshooting what that problem is. Okay, we have obviously, we're going to look at this grossly, we have prokaryotic systems and we have eukaryotic systems. And just because the cells and the tissues are functionally different, structurally different, we use some of the same things, but we kind of approach it a little bit differently. If we're looking at something like a prokaryote, we basically have the large circular genomic DNA, or something like E. coli, which is about 14 million base pairs. A number of the prokaryotes also have native occurring plasmid DNA, which is much, much smaller than the genomic DNA. And so when you are isolating DNA out of one of these prokaryotic kinds of cells, you're going to purify DNA. Some of it's going to be genomic DNA, and some of it's going to be plasmid DNA if there are plasmids in that particular cell system. Now, if it's okay that you co-purify both kinds of DNA, if that's not going to in any way negatively affect your downstream question, your research question, then it's fine. But if it's going to negatively affect it, if you have to have those two DNAs completely separated from each other, then you're going to have to do some extra things. You'll probably never see plasmid DNA in a genomic DNA isolation from a prokaryotic system because you're dealing with DNA molecules that are so different in size. The size of the genomic DNA overwhelms the plasmid DNA that is present. Okay? So, how do we isolate genomic DNA from prokaryotes? Before I go through this quickly, I will say that if your goal is to isolate the plasmid DNA, you absolutely must get rid of the genomic DNA because you'll never see your plasmid if you co-purify genomic DNA with it. Okay, for the genomic DNA, what you do obviously is you culture your cells overnight, you get a whole big bunch of cells, and then you pellet those cells, pour off the media, and then you have a bunch of cells that you can then manipulate and isolate the DNA from them. So what we do is we pellet the cells, and the next thing we do is we lyse the cells. And typically, if you're working with gram-negative bacteria like E. coli or something like that, we just use detergent. One of the more common detergents used to break open the cell is SDS, stands for sodium dodecyl sulfate. It's a workhorse in molecular labs. So we lyse the cell. If you're going with bacteria like bacillus or things like that, we often have to put in an enzyme that will digest away the cell wall. But with something like E. coli, we don't have to do that. The rest of the protocol involves getting rid of the things you don't want and purifying the things you do want. I mean, that's really basically the strategy. And the order in which you do some of these things isn't important. One of the things we would want to get rid of, because it will swamp the system on a gel too, is RNA, because RNA co-purifies with DNA, and DNA co-purifies with RNA, which makes perfect sense because they're both nucleic acids, right? And so they're going to respond the same to the chemicals that we use in the isolation protocol. So we will often hit early on the cell soup, as it were, that's come out of the cell. We often hit it with RNases, which are enzymes that will simply digest away the RNA, and you don't have to deal with it anymore. You are going to be doing a genomic isolation protocol that is identical to this, which is why that's the example I'm using. You're going to be isolating the large chromosomal, in quotes, DNA from a blue-green bacterium. And we're going to clean up the prep with phenol chloroform. So, phenol is caustic, and of course chloroform will put you to sleep. So phenol chloroform is used under the fume hood. And what we do is, when we are at that step, we mix the DNA in whatever solution we have in the phenol chloroform. We mix it carefully to get the phenol chloroform all the way through. And then we spin it in a centrifuge. And what we will get is phase separation. So when you look at your tube, as you get them out of the centrifuge, what you will have are two phases. Your phenol will be down here, and your aqueous layer will be out here. Now the DNA that you are trying to isolate and purify is always in the aqueous layer when you're looking at different kinds of chemicals. What you will also see in this prep is you will see at the interface of the two phases, you will see trying to think of what it's called. It's debris. It's something that you don't want. Basically, it's going to look waxy. It's going to be white and going to look kind of shiny or waxy. And it will, depending upon how much came out, it will sit here, right at that interface. Generally, the first phenol chloroform wash that you do on these isolations, you'll have a pretty good deal of that gunk. And what that gunk is cell wall debris, and things like that. If there are any phenolics, which there aren't in bacteria, that would come out in this layer as well. So what you do after that first spin is we want the layer that has the DNA in it so that we can rewash it with phenol chloroform. Now, as I said, because this is caustic, not only do we use phenol chloroform under the gym hood, but you all need to be gloved when you're working with it, because if you spill it on yourself, it'll burn you. But what we would do then is we would take a pipette and we would suck up the supernatant and transfer it to a new tube. However, what you have to be careful about when you do this is that you don't suck up any of this layer. So the way we get around that as much as possible, because this will just further contaminate, and I'll be washing it until you get rid of this stuff completely. What we do is we put our pipette tip just in the top, and as we are sucking the supernatant up or the top layer off, we move our tip slowly down. So that we always have our tip within liquid, but we're keeping it far away from this zone until you get down near this zone. And usually what we do is we end up leaving a little bit of our supernatant on top of this so that we don't get any of this phenol crap. And you have to be careful, and you have to have really good eyes, and you have to watch what you're doing when you pipette in there. We transfer it over to a new tube. We add fresh phenol chloroform. We mix it, and we centrifuge it again, get another separation. And for some prep, what determines the number of times you wash with phenol chloroform is this thing going away. If the next time you do the phenol chloroform and you have your phase separation and you still have this white layer between, then you'll take the top layer off, put it in another new tube, and you will wash with phenol chloroform again until you do not see it anymore. Okay? Once you've done all of that, you've gotten the DNA out pretty much. The phenol take care of a lot of the proteins as well, get rid of those. So once you've done your phenol chloroform washes a couple times and you have collected your top layer from the final phenol chloroform wash, what we do then is simply precipitate the DNA, and to do that we use alcohol, and most of the time we use isopropanol in this particular step. I told my class in lab at the beginning of the semester that DNA will not stay in solution if there is alcohol around. So if you add alcohol to a DNA solution, the DNA is going to fall right out of solution. Usually what we do is we help this, and this is what you will do in lab in 2 weeks. When you are to the DNA precipitation with isopropanol step, you'll add the alcohol, you'll mix, and you'll put it in the -80 freezer for 30 minutes, and the DNA will just fall out of solution. It's really kind of cool. You won't see this because you're working with smaller DNA molecules, but if you're doing a big prep and you add the isopropanol at that step and watch it, you can actually see DNA precipitate. There's so much DNA. After 30-minute incubation in the microscopic freezer. We spin it down. The DNA will be spun down into a pellet, and you pour off the isopropanol. And then you wash a couple of times with ethanol. Generally, the ethanol that we use for this wash is not 100% ethanol. It's like a 70 or 80% ethanol. And what is the other 30 or 20%? Well, it's just water, okay? And so what you are doing in those washes is salt co-purify with DNA. So when you wash with 70 or 80% ethanol a couple of times in the prep, what you are doing is removing salt because they will dissolve in the water component of the 70 or 80% alcohol. And then you do your final spin, you pour off the alcohol, and then you have to evaporate the alcohol off. Now, we obviously have a speed vac in the lab, but we do not use it for genomic DNA isolation because it overdries the DNA and you can never get it back in solution. And so what we do, and what you will do in a couple of weeks, is that you will pour off the final bit of ethanol and get as much of the ethanol gone as possible, and then you'll just set your tube with the lid open under the fume hood, and it will pull the glass front down so that it's drawing air into the room over the tube, and it will take about 15 minutes to evaporate off the last bit of ethanol, and then the DNA is ready to be dissolved in PE buffers. Okay? So that's the way we do the genomic isolation. Any questions about that? Yes.
So there's no chloroform, is that different or like all of the bits of the cell besides the DNA? I'm wondering what it's getting cleaned up with the cell chloroform.
The DNA itself is getting cleaned up. You're getting rid of the other chemicals. Okay, you don't have many parts of the cell because you've lost the cell over anyway, and this is prokaryotic and any organism. Okay, so you've got a stew of DNA, RNA, and proteins, and pieces of the cell wall. Is basically what it's called. The cell wall and membrane, because membranes are high in lipids, that is what's coming out in this white layer, right there. I'm
fairly sure what we'll be working with won't work, but do you know of organisms that have like native DNA aces, like that'll destroy DNA?
All organisms have those.
So how would you deal with those in an extraction if they couldn't, because couldn't they?
What we do with them is we deal with them early on when you have tissue, which is particularly true of eukaryotes. If you are going to isolate DNA from eukaryotic tissue, and unless you're working with a single cell eukaryote, you're working with tissue. If you're working with eukaryotes, if we stop all metabolic activity, including all enzyme activity, by flash freezing the tissue in liquid nitrogen. Other questions?
Just to clarify, so the top layer will have the chloroform?
Yes, and water. Other questions? Okay. Now, we work a lot with plasmids because we use them as cloning vectors in molecular labs. And plasmid DNA that is not native plasmid, that is bioengineered plasmid DNA. Native plasmids run 60,000 to 120,000 base pairs. Okay. So plasmid DNA, this cloning vector, is much smaller than that. If we don't have much cloned into it or anything cloned into it, those plasmids are going to be about 3,000 base pairs long, 3,000 to 6,000 base pairs. And if you clone something into it, about your maximum size of cloning into it is going to be 10, 12,000 base pairs. So you're still talking about a molecule that is less than 20,000 base pairs. So they're relatively small. You would be overwhelmed isolating plasmid DNA from this prokaryotic cell. You would be overwhelmed by the genomic DNA of the prokaryotic cell. So typically when we're going to do a plasmid isolation, again if you're not using one of the kits, we do what is called a mini prep. And it's called a mini prep for a couple of reasons. One, you're isolating little DNA, mini DNA, and two, you're isolating a small volume, so that's mini. So we just call it a mini prep. And the mini prep protocol is actually beyond clever. I don't know who figured it out, but it is quite clever. Because one of the big things you want to do is to get rid of the genomic DNA. And you can't just go in there with DNases and digest away the genomic DNA, because it'll digest any DNA, including the plasmid DNA that you want. Okay? So what we do is we grow up the cells overnight again, and we collect the cells, we collect a certain volume of cells, put it in an Eppendorf tube, and spin the cells down and pour off the media. Usually what we do is use 2 ml. So we pull up an ml, put it in an Eppendorf tube, spin it down, pour off the media, pull up a second ml, put it on top of the first, spin it down, pour that off, and we've got the number of cells we need to work with. And the next step is, of course, going to be to lyse the cells. And we're going to lyse the cells with SDS, just like we did for the genomic isolation. If you'll notice that there's a kink in this particular thing, we lyse the cells with a mixture of SDS, which is the detergent that will open the cell up, and sodium hydroxide. So we need to ask the question, why are we adding sodium hydroxide? What do you know sodium hydroxide will do to DNA? Well, it is a base, you're absolutely right. So what's it going to do to the pH of the solution?
Raise it.
It's going to raise the pH of the solution. What do we know that raising the pH of the solution is going to do to DNA structure?
Melt the strand.
It will melt the strand. Just like heat will melt the strand, you can melt DNA with sodium hydroxide. So basically, in this step, we're opening up the cell and we're melting the DNA. Well, that doesn't sound like a good idea. So let's see what's going on with this. This slide doesn't want to do anything. I checked this before I turned this on. I think this thing is from this. Quite frankly, I'm sure. Now, this is this. Okay, so let's see what's going on. Here's our bacterial cell, here's our large genomic DNA, it's called the chromosome. And then here are the little plasmids. We're going to hit the cell, and this is what the DNA looks like in here at this point. We're going to hit the cell with SDS, sodium hydroxide. The SDS is opening up the cell wall, cell membrane system. And the sodium hydroxide is raising the pH, and so basically all the DNA is going to melt. Both plasmid and genomic DNA. The other thing that's happening in that tube from the sodium hydroxide, besides the DNA melting, is it starts proteins precipitating. Okay? And so we've got this big melted genomic DNA. And we've got proteins that are starting